Welcome to the second edition of Water Life Support Conference. This is kept by Academy of Agriculture and Forestry Science of Romania, Gheorghe Ionescu-Șișești, and the Food Industry Section, and also the Institute of Research and Development for Aquatic Ecology, Fishing and Aquaculture from Galați, Romania. This conference, it's the second edition. The first one was last year. And the opening of the conference, according to the program, who everybody already have, will be made by Professor Dr. Dumitru Militaru, Vice President of the Romanian Agriculture and Forestry Academy and President of Veterinary Medicine Section.
The participants in this symposium. Deci, încă o dată, bună dimineața! Mulțumesc domnului director Dima pentru invitația de a vă adresa un cuvânt de salut din partea Academiei la începutul, la deschiderea acestui simpozion important pentru dumneavoastră. Bineînțeles că Academia este întotdeauna aproape față de toate institutele noastre de cercetare și față de toate activitățile domnilor lor. Suntem bucuroși să participăm la astfel de manifestări, unde sunt prezentate rezultatele activităților de cercetare și cu toate că nivelul finanțării nu este cel corespunzător, în domeniul dumneavoastră de activitate există rezultate ale activității de cercetare cu care trebuie să ne mândrim și pe care trebuie să le promovăm. Să le promovăm atât prin publicații, dar să le promovăm, mai important, prin acel transfer tehnologic, prin transferul rezultatelor în activitatea de producție. Prin aceasta, vedeți dumneavoastră, încercăm să mai contrabalansăm anumite păreri ale unora, în special din mediul politic, care spun că cercetarea cheltuie bani și nu livrează rezultate. Noi dorim să demonstrăm că livrăm rezultate. De fapt, vis-a-vis de faptul că cercetarea cheltuie bani și nu livrează rezultate, am asistat din punctul meu de vedere la o declarație puțin mai nefericită a unui înalt demnitar din țara noastră, care spunea că trebuie să avem o dezvoltare economică puternică pentru a avea bani pentru cercetare. Da, până aici este bine, dar domnia sa a continuat. Cercetarea nu mai cheltuie bani și nu aduce bani. Iată că cu acest aspect nu am mai fost de acord, pentru că cercetarea făcută așa cum trebuie, abordând tematică prioritară pentru fiecare domeniu, poate să aducă pe termen mediu sau lung înzecit, însutit decât s-a investit. Simpozionul dumneavoastră are o temă foarte, foarte interesantă, apa, suport al vieții. Să știți că eu am pus 2 puncte acestui titlu și am mai continuat cu o perspectivă asupra interconectării lumii vii. Mi s-a părut mie că ar fi mai interesant și mai complet un astfel de titlu. Tema, la prima vedere, ar fi una simplă, dar știm foarte bine că ea nu e simplă, pentru că apa ascunde în ea complexitatea întregii noastre existențe. De multe ori însă noi reducem apa la o simplă formulă chimică sau la o resursă utilitară sau la un simplu bun de consum, dar lucrurile nu stau așa. Noi știm că totul este mai complex, noi știm că apa reprezintă o matrice în care s-a scris și continuă să se scrie întreaga istorie a biologiei pe această planetă. Eu sunt medic veterinar și n-aș vrea să consider că apa este numai un fluid care hidratează celula. Eu vreau să consider că apa reprezintă un ecosistem complex, că apa este un vehicul, că apa, să mă exprim mai plastic, este un teatru de operațiuni pentru cele mai complexe activități biologice. Vedeți dumneavoastră, toată lumea vorbește astăzi de niște subiecte la modă. Vorbim de schimbări climatice, vorbim de poluare, vorbim de epuizarea resurselor, vorbim de reemergența unor patologii. Toată lumea se referă la aceste lucruri. Noi, ca cercetători, ar trebui să ne gândim că aceste aspecte să depășească pragul laboratorilor sau amfiteatrelor. Noi trebuie să ne asigurăm că prin rezultatele activității noastre de cercetare reușim să contribuim cu ceva pozitiv la remedierea acestor probleme deosebite. Nu vreau să lungesc vorba, că am zis că este numai un cuvânt de salut pentru deschiderea simpozionului dumneavoastră, Dar aș vrea să spun că simpozionul, mă gândesc că ar trebui să nu reprezinte numai o serie de prezentări științifice, el ar trebui să fie o platformă de dialog, de sinergii între cercetători și cercetătorii ar trebui să-și lase curiozitatea științifică să-i ghideze și să împărtășim idei care pot să aducă efectiv progres. Nu trebuie însă să uităm niciodată că prin protejarea apei protejăm însăși definiția vieții în toată diversitatea ei uluitoare. Stimați colegi, mi-a făcut plăcere să mă adresez dumneavoastră. Cred că veți avea niște dezbateri fructuoase cu niște concluzii concrete și pe măsură și ca atare vă felicit încă o dată și vă urez multă sănătate. Numai bine!
Thank you very much. And we'll go to the Professor Dr. Engineer Petru Nicoliță, President of the Food Industry Section of the Romanian Agriculture and Forestry Academy, who will tell us about his opinion from the section, because our institute belongs to this section.
Am adresa din partea secției de industrie alimentară a SAS-ului, Academia de Științe Agricole și Silvice, tuturor participanților la acest eveniment științific, atât celor din țară, cât și celor din Belgia, India, Bangladesh, Tunisia și Republica Moldova, care sper sau deja sunt conectați online, probabil, că nu știu. Deci aș vrea să exprim un bine ați venit în limba română, nu știu cum o să traduceți, așa, welcome, probabil, atât deci celor prezenți fizic la sediul Institutului din Galați, cât și celor prezenți online. Doresc să vă urez succes în desfășurarea acestei de-a doua ediție a conferinței științifice APA, suport al vieții, după cea din 27 martie, deci aceeași dată de anul trecut. Începe să se contureze o tradiție și vă doresc din toată inima să o cultivați în continuare, pentru că este ceva cu totul deosebit. Și sunt convins că această urare se va îndeplini, așa va fi cu succes. Aș începe un element referitor la gazda organizatoare, subliniind faptul real și de apreciat că prin activitatea de cercetare, dezvoltare și inovare promovată, prin proiectele de cercetare naționale sau europene sau bilaterale câștigate, derulate și finalizate, prin diseminarea și valorificarea rezultatelor cercetărilor, compartimentele din structura Institutului și conducerea Institutului au imprimat o dinamică puternică de dezvoltare a potențialului de cercetare a Institutului, în primul rând, precum și o creștere a valorii și vizibilității Institutului și a domeniului de cercetare al dumneavoastră, atât în țară cât și în străinătate. Am ținut neapărat să subliniez acest lucru, ceea ce este real și pozitiv și constructiv pentru viitor. Titlul conferinței de astăzi este inspirat și realist și profund valoros, și anume apa, suport al vieții. Eu aș accentua această idee care derivă din titlu, spunând că apa este fundamentul și structura esențială a vieții pe pământ. Argumentând, și cred că mă înscriu în acele câteva minute, argumentând prin următoarele considerații. Și o să enumăr doar 5, cele mai importante. Deși toți suntem conștienți că apa este ceva esențial. Primul argument este că mediul înconjurător de pe planeta noastră cuprinde atmosfera, adică aerul, care conține vapor de apă, deci apă, apa, cu volumul ei imens, nu numai oceanic, de toate felurile, cu o suprafață de 70% din suprafața totală a Pământului, solul, care conține apă, materii organice care conțin apă și diversele forme de viață care au în constituția lor intrinsecă apa. 2. Fitoplanctonul din mediile acvatice produce prin fotosinteză între 50 și 70% din oxigenul existent în atmosferă, adică mult mai mult decât produc toate pădurile și toate vegetațiile de pe suprafața terestră de pe pământ. Urmare a fotosintezei, planctonul, tot din mediul dumneavoastră de activitate, din apă, absorbe dioxidul de carbon și produce oxigenul necesar respirației prin branhii pentru viețuitoarele acvatice și mai produc polizaharide ca și hrană pentru zooplancton și alte viețuitoare acvatice. 3. Apa este componenta structurală, esențială, fundamentală a tot ce înseamnă viață pe planeta noastră. 4. Circuitul complex și spectaculos al apei, și foarte complex și aproape o minunăție, al apei în natură, asigură armonia și echilibru vieții pe pământ, prin asigurarea condițiilor de generare de hrană și oxigen pentru vegetația și fauna terestră, precum și pentru viețuitoarele acvatice. Și în sfârșit, ultimul, și cu asta termin ca argumentare, 5. Apa este un factor esențial în asigurarea condițiilor necesare pentru producerea hranei omului și a viețuitoarelor de orice fel. Altă idee, nu ratez oportunitatea, așa cum am făcut-o întotdeauna, și salut cu această ocazie frumosul și deosebitul oraș Galați, orașul meu natal și al părinților și bunicilor mei, oraș așezat între ape, Dunăre, Prut, Siret și Brateș, oraș cu rezonanțe și rădăcini vechi și adânci în cercetarea de piscicultură, de pescuit și de chimie alimentară în general. În încheiere, doresc mult succes celor care astăzi vor prezenta comunicări științifice, precum și dezbaterilor și discuțiilor care vor urma prezentărilor de lucrări, iar tuturor cercetătorilor Institutului de la Galați și cercetătorilor participanți din alte institute și stațiuni de cercetare și celor din universități, precum și cercetătorilor științifici din cele 5 țări participante din afara țării, mult succes în tot ce faceți, în nobila și frumoasa activitate de cercetare pe care o desfășurați, care are obiectul acesta fundamental și simplu numit APA, apa. Și coincidența, dar nu este întâmplătoare, face ca acum câteva zile, pe 22 martie, să se fi celebrat Ziua Mondială a Apei, care, urmare a conferinței din 1992, conferința ONU pentru mediu și dezvoltare, s-a hotărât să serbeze această zi a apei în fiecare an, iar România are 33 de ani de când sărbătorește acest eveniment, care e cu rezonanțe profunde, nu numai de formă, și de formă pentru cunoaștere, dar și de fondul fondului. Mulțumesc pentru atenție și sunt convins că va fi un succes această a doua acțiune pe această temă importantă. Mulțumesc!
To the opening from Mr. Associate Professor Dr. Engineer Maricel Floricel Dima, General Manager of ICDEAPA Galați.
Doamnelor și domnilor, stimați invitați, distinși cercetători și colaboratori, Este o deosebită onoare și o reală plăcere să vă urez bun venit la această conferință științifică internațională intitulată APA, suport al vieții, o manifestare științifică care reunește specialiști în domeniul nostru de activitate și nu numai, precum și idei inovatoare și o pasiune comună pentru cunoaștere și progres. Astăzi ne aflăm împreună nu doar pentru a prezenta rezultatele și preocupările participanților, ci și pentru a construi legături interdisciplinare solide, capabile să deschidă noi perspective în cercetare. Așa cum se cunoaște, apa reprezintă nu doar o resursă vitală, unică, ci și un subiect de cercetare complex, aflat la intersecția mai multor domenii, științe ale mediului, biologice, chimice, inginerești, etc. Provocările actuale privind gestionarea resurselor de apă, schimbările climatice și protecția ecosistemelor acvatice necesită o abordare integrată și participativă. În acest context, voi realiza o scurtă prezentare a instituției noastre. Institutul de Cercetare și Dezvoltare pentru Ecologia Acvatică, Pescuit și Aquacultură Galați, instituție publică în subordinea Academiei de Științe Agricole și Silvice Gheorghe Ionescu-Șișești, București, fiind unicul institut de ramură din România care derulează programe complexe de cercetare științifică în domeniul acvaculturii, pescuitului, procesării peșterului și altor resurse acvatice vii. Activitatea principală este reprezentată de cercetarea fundamentală și în special cea aplicativă. De asemenea, în cadrul institutului funcționează 3 laboratoare de cercetare și un laborator experimental de dezvoltare care deține o suprafață de 320 de hectare, unde este practicată activitatea de acvacultură, cât și un asolament agropiscicol. Totodată, în cadrul institutului se realizează evaluarea resurselor acvatice vii prin pescuitul în ape interioare și acțiuni de conservare a biodiversității. Actualmente, personalul de cercetare existent este în număr de 38 de salariați, specialiști în cadrul laboratoarelor de cercetare și dezvoltare mai sus menționate. În altă ordine de idei, programul conferinței de astăzi promite abordări captivante, dezbateri constructive și oportunități valoroase de networking. Fiecare lucrare științifică prezentată în cadrul acestei manifestări va contribui la extinderea cunoașterii și va aduce un plus de valoare comunității academice. În final, aș dori să adresez mulțumiri tuturor participanților, organizatorilor și partenerilor care au făcut posibil acest eveniment. Fără implicarea și dedicarea dumneavoastră, acest cadru admirabil nu ar fi existat. Vă încurajez să profitați din plin de această experiență, să împărtășiți idei, să puneți întrebări și să inițiați colaborări care să conducă la eventuale parteneriate. Declar deschisă conferința și vă doresc tuturor mult succes, inspirație și discuții constructive. Vă mulțumesc și succes în continuare!
Romania, from Station of Research and Development for Viticulture and Winemaking, Bujoru. It's about water consumption, a case study from sustainability approach in viticulture and aquaculture. Mihai?
Good morning, everybody. I'm honored to participate at this conference, a second edition. I also was present to the first edition. And first of all, I would like to extend my regards to Director Dima, Professor Militaru, and Professor Niculiță. It's an honor. This study case, let me share. Great. So this is a case study for a sustainable approach in viticulture and aquaculture. My research team is composed from Mihai Vintilă, me, and a professor from Dunărea de Jos University. As we all know, in Romanian agriculture, the demand for water as a resource is on the increase, and despite the fact or the concerning reality that this research is becoming more scarce, and when looking at the reports, the estimation from the National Administration for Romanian Waters shows that the demand for water in agriculture will reach approximately 7 billion cubic meters by 2029. In particular, the pressure on this important resource has been amplified due to the fact that in the years 2022, 23, and 24 were characterized by extreme atmospheric and pedological drought. This was also seen in Bujoru and Galați, but it was confirmed by the National Meteorological Agency. Why did we choose these two apparent different sectors in agriculture? Well, besides the fact that they may seem very different, the truth is that both viticulture and aquaculture need the presence of water in order to develop. So in viticulture, the complex link between water stress and production yields, wine quality, and viticultural characteristics and wine specificity is very dependent on water, and the current trend is to use RDI techniques that can improve the phenolic compound concentrations. For aquaculture branch, the use of RAS or recirculating aquaculture systems have been very important in reducing water consumption and using as a principle the reuse of water in order to maintain the characteristic parameters for the water used, and also this demands a complex filtration system. Unfortunately, Romania is still dominated by earthen basin types in fish farming, and this classic technique ensures over 75% of the national productions. Through the two examples, we'll analyze how the modernization and the use of ecological practices can reduce and manage the use of water as a resource. From a chronological aspect, the two rows of data that we have analyzed are divided firstly from Bujoru Wine and Viticulture Research Development Station, and the second row is obtained from the Institute of Research and Development for Aquatic Ecology, Fishing and Aquaculture in Galați. The analysis also is focused on articles from the academic literature reviews, and also from public institutions dealing with water management. From the analyzed data, we can we also took into account inputs from Romanian INS, and some annual reports from ICDEAPA and the SCDVV Bujoru. From statistical data shows that almost 69% of total fresh water are obtained from different resources is used in the agricultural domain. The other 19% is intended for the industrial sector, and only 12% is intended for functionality of human settlements. When taking a look back through history, we saw that from the 1900s to about 2010, the total water use in these sectors was about 600 or 6 million cubic meters per year, with an increase by 2027. Let me see just a little bit. With an increase to 27 million cubic meters of water, and the forecast for over 50 million cubic meters per year. So the main problem is related to the efficiency of the irrigation network and infrastructure. For Romania, unfortunately, traditional surface irrigation methods are used on a large scale, while efficient methods or modern methods that use sprinkling or drip irrigation are used to a lesser extent, due to the fact that the implementation of these types of systems are quite complex. They need the supervision of specialized engineers, and the cost of the implementation is a little bit higher, but the end results are more important. When taking a look on the statistical analysis for total groundwater input that is mainly used for agriculture, or is the main source of water in our case, in our study, we saw that for the end, in graph 1, for the end of the analyzed period, we see an increase for 2023 and 2024, and also when comparing total groundwater input versus agricultural use, we also see an increase in this use of water. So registering a trend of an increasing trend of in demand for agricultural use. In also FAO data that we synthesized, we also highlights the increase in recent years with sharp increases toward after 2018, as you can see in graph 4, because graph 4 representing the agricultural contribution to water resource stress index. So when taking a look at total water resource stress, we also see that the increasing trend in demand. So especially after 2019, 2018, 2019, and towards 2024. If we analyzed from an efficiency water use point, we also see that the situation is pretty volatile, because the results have been mixed. So water use efficiency, according to Aquastat, when that compares US cost, USD, dollar, that is the main currency for water cost, being dollars, USD. per cubic meter shows that this efficiency registered a spike for 2018, and then it declined slightly with a better result for 2023, and water efficiency for irrigated land is good for 2022 and especially for the end of the interval, but it also oscillates very largely. Now that we talked about a little bit about water cost and what it means, we are now taking a look at sustainable strategies. So in Bujoru, we are implementing conservative agricultural strategies. And some experimental plots that we initiated showed very promising results. And also, when implementing biocircular strategies such as pomace mulch treatments, showed that these experimental plots registered higher economical efficiency for about 66% when compared to traditional techniques, mechanized techniques, and also using conservative strategies had a positive effect on the environment, and surprisingly, the yields that we obtained from the crops was also the same or higher when compared to the plots that used the traditional techniques. For our analysis for the aquaculture sector, the RAS system that was implemented several years back through an operational program showed very promising results when compared to traditional techniques, because the total amount of new water that was used in the production intervals was only 10% when compared to, as I said, to traditional techniques. So optimal use of, when talking about the use of RAS recirculating systems, we also saw an optimal use of reproductive technologies, and from a post-embryonic development to super intensive growth. So the modernized installation optimized the water infrastructures to a drilled well towards 2 storage tanks, and then a water distribution system. Also, to make water use more efficient, the filtration system and water pumping timing was changed, and as a result of these improvements, there was an increase in water quality and reduction in water consumption as a resource, and also, of course, the results in the biological material were obtained. Now, talking about the first steps that we can make towards maybe implementing these strategies and preventing some deficiencies, we need to talk about the simple climate parameter monitoring, and this represents, in many cases, the first line of defense. So the current climate trend shows an increase in frequency for extreme phenomena, mainly characterized by severe atmospheric and pedological drought, with new maximums reached for temperatures that are non-specific for this climate. And also we saw a decrease in the number of days with useful precipitations, and of course this constitutes or amplifies the difficulty for each agricultural branch. In conclusion, our current environmental elements are leading to a future with fewer usable water resources, and these facts can no longer be contested. And of course, new technologies and practices that that are integrated in technological links, as we studied in this study case, are leading towards the obtaining of yields that are comparable to traditional practices or even higher. And of course, the use of artificial intelligence can open new perspectives regarding the sustainability of these two sectors in increasing difficult environment conditions, reaching the goal that agricultural strategies must follow water use decrease and put Romania agriculture towards a sustainable future. Thank you very, very much for your attention.
Thank you very much for presentation. Stop sharing. If our question, no. If not, we will go to Mr. Saber Hamami from National Institute of Agroeconomy of Tunisia. Mr. Hamami, you are online? Okay, we see you. You have a paper about influence of flexible submergent vegetation. Okay, please share to us, and after the presentation, if someone wants to ask you, we can do it. Yeah?
Good morning. Thank you for giving me the opportunity to speak today. Good morning, everybody. I'm Hamami Saber. I'm a researcher from the National Institute of Agronomy of Tunisia. It's an honor to present our work entitled Influence of Flexible Submerged Vegetation on Pollutant Transport in Free Surface Flow Under Continuous Injection, made by me and Madame Hela Rumban and Madame Amel Soulmeyer. Let's start with the outline of this presentation, which begins with a general context. Then we will set the objectives of this work. After this, the experimental study will be presented, and finally we will present the results and discussions, and we finish our presentation with some conclusions and perspectives. Managing water quality in rivers, irrigation canal and drainage networks is a major environmental challenge. The increasing presence of pollutants, even at low concentrations, can have long-term effects on ecosystems, particularly through bioaccumulation. Therefore, it's crucial to understand how these pollutants are transported and dispersed. The context of this work, which focuses on the major and common problems of free surface flow, such as the presence of aquatic vegetation, modifies flow structures, the influence sediment transport and deposition, and ultimately controls the fate of pollutant in water system. So it's already been recognized that excessive plant growth in stream is a bit important as it can significantly affect flow patterns. Indeed, the vegetation represents porosity for flow, significantly change the velocity profile, and modify the transfer and transport of momentum, sediment, and pollutants, and modify the morphology of river beds. So these discharges were generally punctual and can cause various problems which becomes more pronounced with the vegetation presence. The damage of the infrastructure such as hydraulic and road structure, and so river morphology causing overflow and flooding of cities. And also can pose a threat to the global environment, such as a disturbance of the aquatic ecosystems. It is therefore important to know the river capacity to mix and transport these contaminants and to determine the pollutant dispersion rate in solution. On this basis, an experimental analysis work was led concerning pollution propagation in free surface flow at the laboratory channel over a vegetated or smooth bottom. We focuses on continuous injection, which simulates a progressive pollution input in order to better understand the dispersion mechanism and the hydrodynamic parameters controlling pollutant transport. The tracer used was a colored saline solution injected manually and continuously upstream. We used 3 concentration water samples were collected at downstream of the injection point to reconstruct the spatio-temporal evolution of pollutant flume. Turning now to the experimental study, the experiments were carried out in an experimental flume at the INAT laboratory. It is a rectangular open channel having a slope of 1%, and to simulate submerged vegetation, we used flexible element 4 centimeters in height attached to the bed. The vegetation may cover a 4-meter section in the central part of the flume and remains fully submerged during the experiments. We tested 2 bed configurations, a smooth bed without vegetation and a bed covered with vegetation. The experiment concerned the analysis of solution pollutants transport phenomenon in 2 cases, over smooth bed and vegetated bed. To do this, we have concentrated solution tracers and injected in withdrawal operation were carried out through a set of syringes. This is in order to measure the sample concentrations using the laboratory conductimeter. Let's start with the results and discussion. We noticed that for the different cases, the concentration variation was as following. First, a very rapid rise was observed. This is a convective period. The last phase was characterized by a very slow rise, which over time becomes almost constant. This is the diffusive period. On the other hand, the increased flow rate generates a rapid convection period. This is due to the fact that more the velocity flow increases, more the injected quantity diffuses quickly downstream, where the mixture will be completed and stability state is achieved faster. It can also be seen that the vegetation presence on the channel bottom causes a decrease in the pollutant concentration in water. This is due to the fact that the vegetation presence increases the bed shear stress and subsequently will delay the solution propagation in water. A preliminary simulation of this experimental work carried out using Fluent to validate the experimental interpretation. For this, initial conditions must be precised, such as the pollutant initial concentration corresponding to the concentration found in the channel before the injection. Also, the pollutant discharged masses into the channel, as well as the discharge moment must be indicated in the software, as well as the simulation time corresponding to the period for which the monitoring is carried out. At the location where the injection occurs, the pollutant concentration as well as the injection duration must also be indicated for the model. This figure showed that the global trend observed from fluent simulation shows a good agreement with experimental measurements, capturing the same gradual decline in concentration over time. Both approaches confirm the moderating effect of vegetation with a slower decay rate and extended residence time in the vegetated configuration. Minor are attributed to experimental ancestry and the simplified representation of flexible vegetation dynamic in the numerical model. Finally, in conclusion, this work highlights the key effects of vegetation on pollutant transport. First, vegetation clearly reduces concentration peaks while significantly increasing the residence time of pollutants. This means that pollutants remain longer in the system but at lower intensity levels. Second, vegetation modifies transport dynamics by creating that we call trailing effect. Instead of a rapid, sharp propagation, the pollutant spread more gradually over time. In addition, vegetation acts as a natural filter. It temporarily retains pollutants within the canopy, which helps mitigate the impact of accidentally discharges in natural system. And thank you for your attention.
Thank you very much. I have a small question. I saw the presentation and I saw you have experiment in the area. Do you have also the project in the field to implement something like that in the way to decrease the pollution and to keep this kind of pollutant in the some area?
Yes, we simulate a real case in Medjugorje River, and we have to now it etudiates a real case and show the real propagation in the Medjugorje rivers to know in nature how this can affect river and dams.
Okay, so you build a dam with this kind of stuff, or only a small area of the river?
Only a small area.
Only a small area, because I think if you make a dam with all suffers of the river, and the river can go through this vegetation, after can be a little bit cleaner. No, this was idea.
Yes. Yes. Thank you.
Okay, if are other questions.
Hello, can I ask you a question? You said that you are using vegetation, but is your study, or did you study the use of other sorbents in order to reduce the pollutants present in your experimental plot? I was thinking maybe other sorbents of natural origin. You know, there are studies that use maybe some rocks and so on that aren't specific to your experimental plot. You know, because I was studying the literature, and there are many categories of sorbents used that in order to prevent the peaks in pollutants, and also maybe I was thinking if you did a screening of the microbiota present in the river, and if there are some differences or fluctuations in your experimental plot, as I was, let's name it like that, an experimental plot, because it was a section of the river.
Yes, in reality, we etudiate the dispersion coefficient that is necessary to have idea of mixture of this pollutant. We can etudiate the other effect. The results show that the vegetation affect the dispersion coefficient, and we etudiate this case in experimental channel and in real river. And thank you.
Okay. Thank you very much.
Thank you very much.
If are not other question, we'll go to the next one. Next one, it's from India, Anu Shanduhardi.
Oxidative stress as an indicator of environmental pollution in aquatic ecosystems. Introduction. Aquatic ecosystems such as rivers, lakes, ponds support a wide variety of life forms. However, these ecosystems are increasingly affected by environmental pollution caused by human activities such as industrial discharge, agricultural runoff, and urban waste. Pollutants entering bodies can disturb the natural balance of aquatic organisms. Environmental pollution in aquatic system. Water pollution occurs when harmful substances enter aquatic environments. Common pollutants include heavy metals, pesticides, fertilizers, industrial chemicals, and untreated sewage. These substances can accumulate in water and sediments, affecting aquatic organisms directly or indirectly. What is oxidative stress? Oxidative stress occurs when the production of reactive oxygen species, ROS, becomes greater than the organism's antioxidant defense system. Their reactive molecules can damage important cellular components, such as lipids, proteins, and DNA. Sources of reactive oxygen species. Reactive oxygen species and produced naturally during metabolic process. Environmental pollutants, such as heavy metals, pesticides, hydrocarbons, and industrial chemicals, can significantly increase their production in aquatic organisms. Effects of oxidative stress. Excessive oxidative stress can cause several harmful effects, including cell damage, lipid peroxidation, enzyme inactivation, DNA damage, and impaired growth. In severe cases, it may lead to reduced survival and reproductive failure in aquatic species. Antioxidant defense system. Aquatic organisms process natural antioxidant defense mechanism that protect cells from oxidative damage. Important antioxidant enzymes include superoxide dismutase, SOD, catalase, CAT, and glutathione peroxidase, GPX. Biomarkers of oxidative stress. Biomarkers are measurable biological resources that indicate exposure to pollutants. Common oxidative stress biomarkers include SOD, CAT, GPX activity, and lipid peroxidation levels such as malondialdehyde, MDA. Fish as bioindicators. Fish are widely used as boundaries of aquatic pollution because they are sensitive to environmental changes. Measuring antioxidant enzymes in fish tissues helps scientists to understand the level of pollution in water bodies. Advantages of using oxidative stress biomarkers. Oxidative stress biomarkers provide early warning signals of pollution before visible ecological damage occurs. They are sensitive, measurable, and useful for monitoring environmental health. Research methods. Researchers collect fish samples from different aquatic locations. Tissues such as liver, gills, and muscles are analyzed in laboratories to measure antioxidant enzymes' activity and oxidative damages. Environmental importance. Monitoring oxidative stress helps scientists evaluate the impact of pollutants on aquatic organisms and ecosystems. It also assists in developing strategies for environmental protection and pollution control. Sources of water pollution. Prevention and control. Proper waste treatment before discharge. Reduce use of harmful chemicals. Regular monitoring of water quality. Awareness and strict environmental laws. Conclusion. Oxidative stress is an important biological indicator of environmental pollution in aquatic organisms. Studying antioxidant responses in aquatic organisms helps detect pollution early and supports efforts to protect water resources and biodiversity. Thank you.
Next one, it's from Mohamed Bayrem Hormia. If are online.
Hello, yes, I'm online.
Okay. Can you show your presentation, please?
Yes.
Okay.
Do you see it?
Yeah, perfectly. You can start to explain what is there. Thank you.
So, good morning, honorable members of the committee and guests. My name is Hormia Mohamed Bayram. I am a doctoral student at the National Institute of Agronomic of Tunisia within the Green Team Laboratory. It's a great honor to be here to present our research entitled Extreme Flood Dynamics and Vulnerability of Semi-Arid Plains, 2D Hydrodynamic Modeling in the Majorda Basin, made by me, Saber Hamami, Slim Hossein Talbi, and Amal Swalmaya. So to guide you through our work, my presentation is structured as follows: introduction, study area, hydrological data, hydrodynamic modeling and calibration, and finally results. And finally conclusion. Sorry. Starting with the introduction, it is important to emphasize that agricultural plains located in semi-arid regions are highly fragile ecosystems. They are particularly exposed to extreme hydrological hazard. A prime example of this vulnerability is the Boussel Plain in Tunisia, which frequently experience severe and recurrent flood events from the Majorda River directly threatening the surrounding lands. So our objective is to evaluate the physical impact of 100-year flood event on the agricultural plains by assessing the maximum inundation extent and submergence depth, and analyzing the flow velocity dynamics across the floodplain. Our study area is located in northwestern of Tunisia, specifically in the city of Boussel. This city is part of the watershed of Majorda and is directly traversed by Majorda River. The local topography is characterized by a low and nearly uniform slope around 0.03%. For the hydrological data, we adopt the historical hydrograph of 1973 flood events. It corresponds to 100-year return period with a peak discharge of 3,180 cubic meters per second. To simulate the spatial propagation of this extreme flood wave, the HECRAS 2D was employed with a high-resolution mesh. The hydrograph was precisely routed across the complex topography. The 2D hydraulic model was calibrated by simulating the flood event of 2019. The reliability of this simulation was confirmed through a twofold validation approach. First, simulated water depth were cross-referenced with the actual field observation. During peak discharge of 250 cubic meters per second, the simulated level of 8.14 meters closely matched the observed of 8.4 meters, and for the flow of 180 cubic meters per second was 7.36 simulated against 7.52 meters observed. Second, statistical comparison against the hydrometric station rating curve was conducted. This comprehensive comparison yielded excellent metrics, notably Nash Sutcliffe efficiency of 0.96 and R squared of 0.99. Following the successful calibration, the Manning roughness coefficient were established as follows. For the main channel is 0.033. For riverbanks, it's up from 0.05 to 0.09. For agricultural floodplain, 0.05. And for the urban area, 0.2. Yeah. For our result, the inundation analysis reveals an almost complete submerge of agricultural parcel once the discharge exceeds the main channel capacity, with a water level can almost reaching 2 meters. For the spatial distribution of velocities, the spatial distribution of velocity reveals major contrast. Inside the main channel, flow speed exceeds 4 meters per second. However, once the water reaches the plain, it drops drastically to near zero values, below 0.1 meter per second. This confirms a state of extensive water stagnation because the terrain is so flat, the water cannot drain efficiently. In conclusion, our research proves that the vulnerability of the floodplain is fundamentally driven by flow dynamics, not merely by inundation extent. We demonstrate that the abrupt flow deceleration causes prolonged water stagnation, threatening agricultural soil structure. Looking forward, this finding highlight crucial perspective for regional planning. It is now essential to evaluate the impact of future hydraulics development on flow dynamics by integrating stagnation risk analysis into regional flood management strategies, and we can develop targeted mitigation measures to protect soil structure in highly vulnerable zones. Thanks for your attention.
Thank you very much. If okay, you can. Okay, if there are questions about this presentation, Mihai, please.
Hello. I was just wondering if the results obtained from your study can be generalized towards other areas. I think the results for your study are very interesting, and that's why I was wondering if you can generalize the results obtained in your study. Thank you.
So this was for a semi-arid area. This particular study area is have topography that is so flat. Well, we can. Okay.
I
So-
think,
So-
sorry,
So as
I
a
think,
conclusion,
yeah.
As a conclusion, if we are talking about a semi-arid area, because in Romania we also have these types of terrains, so if we replicate exactly the conditions for your study, the results obtained could be similar, correctly?
Maybe for the
Correct?
mining profession,
Mhm.
that could be similar. The
Mhm.
mining
Okay.
profession.
Okay. Okay. Thank you very much for your response.
Other question?
Yeah, hello. My name is Mark Ionuț, from Institute of Aquaculture. I have only one question for you. In your material, you present some maps. For this map, for creating this map, do you use intelligence artificial, or do you use the classic maps of Google, of another soft for creating this maps? I don't know if
No,
you
the maps
understand
was
my
created
question.
with the QGIS. Uh, I created the maps by myself, I mean.
Can you repeat? Sorry.
The maps was created with the QGIS, with the GIS.
With the G.
So I created the maps with myself.
Okay, yourself. So the classic method.
The
Okay, thank you.
the classic model, yes, it is.
Okay, thank you.
Okay, thank you very much. We'll go a little bit with another presentation from Nucet Farming Research and Development Station from Romania. Will be present probably Marinela Gancea, are online.
Oops. Okay. Good morning, ladies and gentlemen. My name is Marika Nina, and I will present the scientific paper entitled Monitoring Environmental Conditions as a Tool for the Conservation of Fish Genetic Resources at the at the Research and Development Station for Fisheries, Nucet. Aquaculture is one of the most dynamic sectors of the global food industry. Its sustainability is threatened by the degradation of freshwater ecosystem due to the impact of climate change. In this context, the conservation of fish genetic resources becomes a priority for maintaining aquatic biodiversity. The fish gene bank from Nucet Station is a valuable reservoir of genetic diversity used in research and breeding programs for fish species. Nucet Station manages over 30 genetic entities unique in the country, including valuable species such as common carp, Polydon spatula, Asian cyprinids, and predatory species. These resources are both economic and scientific importance. The role of water in protecting fish stocks. Water is not merely a habitat but an essential factor in maintaining the physical and chemical balance of the aquatic ecosystem. It directly contributes to the conservation and protection of fish genetic resources. It provides protection against the biological vulnerability of fish species and the dynamics of environmental factors. The aim of this research was to assess the environmental conditions in the wintering ponds to maintain the physiological integrity of valuable fish species of the Nucet Station gene bank. The study focused on the stability of the water's physico-chemical parameters and their role in the conservation of fish genetic resources. Managing aquatic environments during the winter it is essential for preserving fish genetic diversity. The study was conducted adnotestation in two wintering ponds, which share similar characteristics. Monitoring was conducted over two consecutive cold seasons between November and March, covering the entire wintering period. Stocking was carried out in accordance with technical guidelines based on species, age, size, and genetic value. The stocking structure of wintering ponds was made up of the following species: common carp, Asian cyprinids, and Polyodon spatula, with a stocking density of 7 tons per hectare. The determination of the water's physico-chemical parameters were performed using portable field equipment and standard laboratory methods. Water samples were collected at a depth of approximately 30 to 50 centimeters below the water surface, a level considered representative for assessing conditions in the upper layer of the water column during the winter period. The physical parameters of the pond water were measured daily, and the chemical parameters were analyzed monthly. The ponds were continuously monitored using technical means to assess the water level in the pond, the thickness of the ice layer, and the creation of aeration zones. Assessing the stability of the aquatic ecosystem is essential for preventing critical situations associated with hypoxia or extreme temperature fluctuations. Seasonal variation of water and air temperature. In both seasons analyzed, it was observed that fluctuations in water temperature were smaller than those in air temperature. In March 2025, the largest increase in water temperature occurs, which implies a shorter cold season. In January 2026, the lowest air temperature is recorded. The first cold season was slightly colder than the second season, especially in terms of air temperature, while water temperature showed smaller variations. Assessment of the dynamics of dissolved oxygen concentration. The solubility of oxygen is inversely proportional to the water temperature. The concentration of dissolved oxygen was high in both ponds during the two consecutive cold seasons without reaching critical levels. During the first cold season, the months of November and March recorded average dissolved oxygen concentration of 7.7 milligrams per liter. In second cold season, there was a steady increase in dissolved oxygen concentration values from 9.6 to 13 milligrams per liter. Dissolved oxygen concentration was high through the entire period analyzed. Physical-chemical water quality parameters. The chemical parameters remained within near optimal ranges during both cold seasons. Critical parameters are well below the alert thresholds, confirming the effectiveness of maintaining an optimal flow rate. pH stability suggests a well-balanced ecosystem. The second cold season is characterized by superior chemical stability and better oxygenation. In conclusion, the stability of water quality parameters ensured optimal conditions for the maintaining fish stocks during the wintering period. Conditions specific to the cold season supported the viability of biological stocks. Continuous monitoring directly contributed to maintaining the balance of the aquaculture ecosystem. Proper management of the aquatic environment is an essential factor for the wintering of fish genetic resources. And water quality is fundamental to the conservation and sustainable use of fish genetic resources. Thank you very much for your attention.
Thank you.
Welcome.
If are questions? If are not, we'll go to the next one. Thank you very much. We'll go to the next one from Republic of Moldavia, Mr. Dumitru Bulat, if it's online.
Voi relata cu privire la invazia sângerului în râul Prut, respectiv ca introducere vreau să menționez că sângerul este o specie foarte răspândită în policultura noastră autohtonă din cauza anumitor avantaje, preț accesibil, creștere rapidă și, desigur, sunt anumite economii ce țin de furaje, pentru că nu se cheltuie mult la furajare. Sângerul a fost introdus în anul 1961 în Republica Moldova sub formă de puiet, 100.000 de bucăți de puiet direct din bazinul Amurului. Dar trebuie să facem deosebire între avantajele în ecosistemele cu destinație piscicolă, adică vorbim de hileștei, iazuri, lacuri de acumulare, și în ecosistemele naturale, unde noi trebuie să mergem de pe concepția purității, da, biodiversității și însăși a ictiogeonofondului autohton. Ce ține de râul Prut, sângerul în anii 70, 80, 90 practic nu se întâlnea în râul Prut în capturi, apoi brusc a crescut ponderea acestuia, mai ales după inundațiile majore din 2008 și 2010. Ponderea sa a crescut până la 30% în 2011 și după asta se observă oarecare stabilizare și iarăși în ultimul timp observăm creșterea efectivă a acestei specii. În grupele de vârstă, comparativ cu alte ecosisteme, se observă oarecare ultimul timp continuitate, adică se identifică 10 grupe de vârstă și totodată trebuie menționat că la noi, practic, din speciile de talie mare și însăși exemplarele de talie mare, nu avem în afară de sângere. Ultimul timp, în plasele care sunt date în derivă, da, cu mărimea ochiului de la 80 până la 100, sângerul este o specie iodominantă. În 80 pe 80 avem 76,47% și chiar se constată o abundență foarte mare a speciei în anumite perioade, sunt valori procentuale până la 100%. Am făcut investigațiile pe parcursul întregului an și s-a observat că în trecut, dacă se vorbea despre specii introducente, în ultimul timp deja putem vorbi de o specie naturalizată. Astfel, pe 13 și 14 august au fost identificate exemplare de sânger care se aflau în stadiul 4-5 de maturizare a ovarelor și la care erau identificate membrane foliculare eliberate din prima generație, ceea ce indică că au fost cazuri de reproducere naturală. Totodată trebuie de menționat că sângerul are o etologie foarte interesantă în râul Prut. El vine primăvara din Dunăre, rămâne aici până în toamnă, exemplarele undeva 80-90% după abundență iarăși se răstogolește, coboare în Dunăre, iar o parte mică rămâne în Prut. Și iată s-a constatat că la exemplarele care rămân în Prut, la iernare, nu sunt eliberate toate porțile de ovocite, iar în mare parte se constată resorția totală a acestora în stadiul de creștere trofoplasmatică. Iată, această fotografie este făcută ieri și observăm sângerul din Prut care rămâne, că noi am făcut investigații data asta, am făcut nu în zona Gheorghilești, nu în zona, adică în sectorul terminal al prutului inferior, dar mai sus, mai înspre Costești stâncă. Observăm sângeri care totuși nu s-a reprodus, ceea ce este foarte interesant, pentru că avem un fel de subpopulație care rămâne, o subpopulație în râu Prut și nu se reproduce, și subpopulație care se reproduce, după asta coboară în Dunăre și respectiv iarăși migrează în primăvară în râu Prut. Este o enigmă pentru noi și chiar este o biologie a reproducerii foarte interesantă și desigur noi vom continua cercetările. Am înaintat anumite propuneri la Ministerul Mediului pentru a reglementa pescuitul acestei specii în râul Prut, pentru că este o specie foarte numeroasă. Mai mult ca atât, noi știm că în România se permite pescuitul acestor specii în ecosistemele naturale fără limită, da? Doar cu condiția că să fie înregistrate la Agenția Națională pentru Aquacultură și Pescuit. Noi am propus să se modifice legea 149, unde să se elimine acele 5 kg permise, maxim de 5 kg permise și pentru specii de pește pe zi și de asemenea unele specii care vor fi declarate ca specii alogene invasive, iar să fie capturate fără limite. Astfel, la noi este în curs de aprobare în a doua lectură legea cu privire la speciile invazive, unde ne propunem ca sângerul în râu Prut să fie considerat o specie alogenă cu efect invaziv. Și desigur vor fi și numeroase beneficii, pentru că este o specie totuși care se mănâncă la noi în Moldova și în acest aspect va fi într-un fel sau altul asigurată securitatea alimentară a țării și de asemenea și în aspect ecologic iarăși vom avea o serie de beneficii. Dacă sunt întrebări, cu plăcere vă răspund la...
Cercetător științific, coordonator la Institutul Național de Cercetare Aplicativă în Agricultură și Medicină Veterinară din Republica Moldova. Studiul nostru vizează diversitatea parazitofaunii și particularitățile ichtioparazitogenozelor la pești din bazinele piscicole a Republicii Moldova, în contextul degradării ecosistemelor acvatice și al dezvoltării acvaculturii. În urma cercetării, noi am identificat 168 de specii de paraziți, aparținând la 15 încrencături taxonomice, cu predominație cealetelor, cnidiosporidelor și helmințelor. Cele mai afectate specii au fost, din cercetările noastre, cele mai afectate specii caras și novac. Speciile de caras și novac au avut o prezență rezistentă mai ridicată la paraziți. Rezultatele indicate au fost intensificarea pisciculturii și degradării calității apei favorizează apariția paraziților cu impact episodic, în special la puieți. De asemenea, structura comunităților parazitare este influențată direct de factorii de mediu, cum ar fi infecția apă și altele. În concluzie, paraziții, în special cnidiosporidele, pot servi drept ca bioindicatori ai stărilor ecosistemelor acvatice, iar monitorizarea acestora este esențială pentru prevenirea epizotilor și pentru un management durabil al resurselor piscicole. Foarte succint v-am prezentat ceea ce am redat în poster și eu vă mulțumesc foarte mult pentru atenție.
My name is Angelica Dobre. I'm with the Institute of Research and Development for Aquatic Ecology, Fishing and Aquaculture from Galați. And I'm going to present the paper entitled Body Condition Dynamics of Pontic Shad During Spawning Migration in the Lower Danube between 2020 and 2025. Let me share the presentation. Okay. Can we see it?
Yeah, perfectly.
Okay. Okay. The Pontic shad is an anadromous species of major ecological and commercial importance for the Danube-Black Sea basin. Its life cycles involves a marine feeding phase in the Black Sea, followed by an upstream migration into the Danube River for reproduction. Historically, this migration extended up to 1,000 kilometers, but today it's significantly restricted due to hydropower dams, overfishing, pollution, and altered hydrological regimes. Spawning migration represents a high energetic cost period. During this phase, fish condition is influenced by water level, temperature, habitat accessibility, and food availability. To assess the physiological state, we use the Fulton condition factors, which reflect somatic energy reserves, nutritional condition, and overall physiological status. The main objectives was to evaluate the temporal evolution of body condition during spawning migration. The study was conducted in the lower Danube sector between river kilometer 133 to 197 during March to June, over the period from 2020 to 2025, corresponding to the spawning migration period. We analyzed 2,130 individuals, and for each we measured total length and weight. The analyzed individuals had an average length of 30 centimeters and corresponded to adult migrating fish, indicating that the analyzed samples mainly consisted of reproductively active fish. The sex distribution consisted of 56.7% of females, while the rest of 43.3% males. This female predominance could reflect migration timing differences, behavioral differences during the spawning migration, or possible sampling selectivity. To better understand the temporal changes in body condition, we examine its interannual variation across the studied period. The statistical analysis results showed significant differences between years in both sexes, showing that body condition was not stable across years, possibly due to environmental variability influencing energetic status. Separately, both females and males showed strong interannual variability in body condition. Moreover, in female, there was a progressive decline in somatic condition, while males peaked in 2023, followed also by decrease. The sex-specific temporal patterns suggest differential sensitivity to environmental influences. Across all years, female consistently showed higher K values than males. Given the higher energetic demands associated with vitellogenesis and oocyte production, females are expected to respond more strongly to hydroclimatic constraints affecting migration efficiency. Regarding the hydrological influence on body condition, a positive relation was observed between river water level and body condition, with a moderate correlation in males and a strong correlation in females, suggesting that females are likely more sensitive to level dynamics that influence migration efficiency, spawning synchronization, and energy allocation. This sex-specific sensitivity supports the hypothesis that hydrological conditions represent a critical environmental driver of reproductive energetic status during spawning migration. Hydrological variability affects migration efficiency, spawning synchronization, and energy allocation. Females show stronger responses due to vitellogenesis and higher energetic demands. Therefore, it directly influences reproductive success, recruitment, and population stability. Hydrological regime alteration may indirectly affect population dynamics of Pontic shad. In conclusion, Pontic shad body conditions show significantly interannual variability. Sex-specific responses were clearly observed. Hydrological conditions are a key driver of energetic status, with female exhibiting greater ecologic sensitivity. Maintaining appropriate hydrological regime is essential for sustaining reproduction and long-term population resilience. These results highlight the importance of integrating hydroclimatic variability into fishery management strategies for migratory species such as Pontic shad. Thank you.
Thank you very much. Alexander Moreau, Belgium. Alexander, you are online?
Yes, hello everyone.
Okay, you can
Do you hear
start
me?
your presentation. Thank you to be present.
Thank you for the invitation. Good morning, everyone. I hope the connection is it's good, and feel free to let me know if at any moment you don't hear me. Okay,
Yeah,
I'm sharing my
it's
screen.
good. Full screen. Perfect. You can start now.
Thank you. Thank you, Mihai. Much appreciated. So yeah, basically the intervention of today. So I'm Alexandre Moreau. I'm the managing director of Advagreen. Advagreen, we are a Belgium company, expert in the formulation and the production of methylene urea molecules. So basically, and we will get into the details, but we are producing methylene urea molecules, which are applied as a fertilizer on various crops. Here, there are a lot of things to say about those molecules, as you may imagine. Here for this intervention, we of course focused on how methylene urea molecules may reduce the water ground, reduce the pollution of the water ground. And this will be let's say, the core focus of this intervention today. Basically, just a bit of context before jumping into why methylene urea molecules might be of interest on this problematic. It's always interesting to get back on what is the current situation today with the main fertilizers which are used. I'm convinced that here I'm learning nothing to anyone, but it's always a good base to get back to this observation from the universities. Long story short, and you see on this graph, but when we look at what type of fertilizers is mainly used in our farming industry today, we're going to speak about urea when we want to supply nitrogen. It's the most concentrated one. It's the one which is the most on the market, even if it's shaking a bit right now due to other reasons. But farmers are going to apply urea. And then the main problematic is going to be that there is no synergy between the nitrogen released from the urea that the farmer is going to apply with let's say, the assimilation curves of the crop. In other way, we're going to apply a nitrogen fertilizer, which is going to release into nitrate quite fast. And the nitrate not being stable, we're going to have a lot of losses through lixiviation or volatilization. We're going to focus even more now on the lixiviation because we link it to the water, but we have a lot of losses. Again, the numbers that agronomists are agreed on, even it's always a precise and tricky part, and there are a lot of counterexamples, but 40% losses from the nitrogen brought through urea. So that's a bit the context, and we have a problem on the lixiviation and then on the water pollution. If I can share a story, I'm from France, even if the company is in Britain. And we do have a lot this problem in Brittany because we use a lot of those fertilizers for the maize, for instance, and then it goes in the water, and then we have the algues vertes, we call them like that, the green algae, which are overdeveloping on the sea due to this problem. So there is a clearly important problem, environmentally speaking. So how do we approach this problematic, and how do we bring a solution, partial solution as well? There is no miracle, so we attach one part of this big problem But with what we develop, with those methylenurea molecules that you do see on the screen right now, before getting into the detail of the chemical part, what we are doing is that we are creating in our reactors, in our processes, complex. Why is it important? Because they will take more time to be released into the ammonium form, to be released into the nitrate form. So having those more complex molecule, which are going to progressively release the nitrogen throughout and up to the nitrate phase, it's going to allow the crop to absorb the nitrogen progressively. In other words, when we look at the urea having a release of, let's say, 1 week maximum after application, when the farmer is going to apply methylenurea, there is a progressive release in between 4 to 6 weeks, depending on some pedoclimatic conditions, such as the temperature, such as the moisture, such as the quantity of light. And that's how we do address this problematic. Basically, it's to say that we must bring a nitrogen form which is going to be more stable, and that's what triazones, that's what you see those three yellow molecules, the MMU for the monomethyllurea, the DMU for the dimethyllurea, and the triazones. They are stable. They are stable, and they will take time to be break down into the nitrogen forms that you do know, the urea, the ammonium, and the nitrates. And that's how we approach this problematic, and that's how we partially solve it, as I mentioned, because as it's a liquid product, we apply it on certain stage, not on all of them. But when we can target a leaf application, then using those types of molecules, it's clearly bringing an improvement regarding the reduction of the nitrogen losses overall, and it's in particular in the nitrogen losses in the water through the leaching. Let's be a bit more scientific and to measure it. And there is actually quite an interesting article, which is that you do have on the screen right there. It's basically showing the quantity of nitrogen which is lost through nitrate form, NO3-. And what do we see here is that we are bringing a clearly improvement when using methylenurea molecules compared to the standard. We based on the standard, which is the urea one. And when we check the urea, you see there are different depths, small one, medium, or deep one. In any case, in all the scenarios, using the methylenurea is going to decrease significantly the quantity of nitrogen which is going to lose in the water. So it demonstrates somehow that using such molecules will really bring first an environmental benefits for our water ground. We are going to pollute it less with the nitrogen that farmers are applying. And it's also, I mentioned it because it's important for farmers today, bring an economical improvement, because what they're going to apply is going to be absorbed and transformed into yield and quality, yield and protein, for instance, on a wheat. And that's the conclusion slide here. That we do reduce, it's going to be repeating, but it's important. We do reduce through using methylenurea molecules the risk of leaching due to the fact that we have those complex molecules, the monomethylol, the dimethylol, the triazones, which are going to take more time to be released. The fact that they're going to take more time to be released into an absorbable form by the crop, and then it will increase the quantity of nitrogen assimilated and reducing by consequence the losses through volatilization or leaching. They asked me to do like 5 to 10 minutes, so I've tried to be straight to the point. Do you have any question or remark?
If someone have a question. We saw you make some magical with the water to keep the water in the molecule and release the water in time. This is all chemical stuff we told us.
Exactly. So it's not a sponge neither. There are some products, actually. I was studying one in Iran. They developed a molecule which are going to be water retention. So here with the methylenurea, we remain on the fertilizer part. Nevertheless, indeed, it plays the sponge one, because basically we can take this image of charging the sponge with nitrogen, and then we press it progressively so the nitrogen gets out of the sponge, you know, over 4 to 6 weeks. And that's the methylenuria. Thanks for the image. I'm going to use it again.
Okay, it's like not like Bob Sponge, but something similar.
We can give him the nickname. It's okay.
Okay, if there are other questions. Before that,
this is not a question, is an appreciation. Alexander is a very good partner to have, and I'm glad that we are collaborating and doing interesting researches regarding the use of slow release fertilizers, and these types of studies, I think, are paving the way towards a sustainable future in agriculture. And yeah, I'm glad to see Alexander again. We talk very often, and I'm really glad that we are collaborating. Thank you very much.
Go to the estimate total biomass of Hirudinea across 5 lakes of the Matia Merdhei complex from the Danube River in 2025. Decimira?
Yes.
Okay, start.
Hello. Thank you. I will share the presentation. You see it? The presentation?
Yeah, it's okay. Start.
Thank you. Hello, everyone. I'm presenting our study on the estimated total biomass of the Hirudo verbana, commonly known as the medicinal leech, in the Matița Mehei lake complex of the Danube Delta. While the medicinal leeches are a vital medicinal resource, they are also a species of conservation concern. Until now, most studies were descriptive and local, lacking a system-level perspective on how much biomass actually exists at an ecosystem scale. So our main goals were to quantify the total biomass, assessing the spatial distribution across 5 specific lakes, and understand the relationship between the lake area and biomass productivity. We focus on the 5 lakes: Alb, Meșter, Martin, Topolga, and Carasu. We specifically looked at about 30% of the lake surface, which represent the suitable habitat for this species. So we used 10 control plots per lake. The methods involve fixed point water disturbance to attract the leeches. Biomass was then calculated based on the mean, the density, and individual mass using log regression and Poisson approximation for the uncertainty. So our feedings show a total estimated biomass at 18.59 tons. However, there is a high variability in the sites. So, okay, takeaway is that the biomass is not evenly spread. In fact, 82% of the total biomass is concerning just in the 3 lakes: Alb, 30.2%, Martin, 26.1%, and Meșter, 25.8%. we found a sublinear scaling relationship exponent B are 0.627. This means that as the lake gets larger, that the biomass doesn't increase proportionally, it's slowed down. Interesting smaller lakes like Carasu and Topolga showed a higher aerial efficiency kilogram per hectare. Essentially, smaller lakes are more productive per units of area. So to conclude this study, the biomass of Hirudo verbana is highly structured and aggregated. Smaller lakes are hot spots for productivity. About management implication for sustainable management, harvesting should not be uniform, because smaller lakes are more productive, but also more vulnerable of overexploitation. We recommended a spatially differentiated harvesting strategy to protect this population. Thank you for your attention.
I will present you the role of water in modulating functional mechanisms underlying algal ecophysiology. Pollutants are defined as solid, liquid, or gaseous substances that at certain concentrations become harmful to ecosystems. The major type of pollution, air, water, and soil pollution, each having specific impacts on the environment. The role of human activities such as urbanization and industrial effluents is coming in water resources. Environmental stress affects organisms at different levels, from cells to entire ecosystems. In the short term, organisms may adapt and cope with these stress factors. However, when the stress is strong or lasts for a long time, it can exceed the organism's ability to maintain internal balance, leading to dysfunction and negative effects on health and survival. Generally speaking, the characteristics of water and the final intended use of water determine the degree of treatment and the treatment method. Mostly the focus is on water treatment plants that produce safe drinking water, and wastewater treatment plants that treat water for safe discharge or future reuse. This slide illustrates the multifaceted impact of chemical pollutants on aquatic ecosystem, emphasizing their origin, bioavailability, and toxicological consequences as industrial emissions, biomedical waste, and agricultural inputs like pesticides and insecticides introduce a complex mixture of contaminants, including heavy metals and organic xenobiotics into aquatic environments. This slide provides a comprehensive overview of the primary source and pathways of environmental pollution, with particular emphasis on the integration of anthropogenic activities into aquatic system. Domestic sewage, industrial effluents, and agricultural discharges represent major point and non-point source, introducing a diverse contaminants, including heavy metals, persistent organic pollutants, and synthetic polymers. These substances are often resistant to degradation, thereby contributing to long-term environmental persistence and ecological risk. Environmental stress factors affect living organisms in multiple ways. So a broader approach is needed to understand the change that occur in the molecular level. At the cellular and physiological levels, respiratory stress is one of the first responses, especially in aquatic organisms. When the members involved in respiration are damaged, the diffusion of oxygen and the exchange of ions are disrupted. Biodegradation of toxic compounds is natural ability of microorganisms to metabolize chemical substances, converting them into less harmful or toxic compounds. This procedure is considered an environmentally friendly and economically viable strategy for decontaminating water affected by pollutants. Several microorganisms have been identified with the ability to decompose hydrocarbon. For example, microorganisms like Pseudomonas, Bacillus, Micrococcus, or Acinetobacter can degrade petroleum and aromatic hydrocarbons based on the source of carbon and ambiental temperature. These species are representative, but there are others with similar capabilities. Water treatment involves a combination of physical, chemical, and biological processes aimed to modifying water quality to meet specific end-use requirements. The selection and intensity of treatment depend on the intended application, water for drinking, industrial use, irrigation, recreational purposes, or safe environmental discharge. The mechanism of microbial biodegradation are highly dependent on the metabolic capability of the microbial community and the chemical structure of the pollutant. Aerobic biodegradation, release on oxygen as terminal electron acceptor, and in particularly effective for organic pollutants such as polycyclic aromatic hydrocarbons, phenolic compounds, and pesticides. Anaerobic pathways, by contrast, operate in oxygen limit environments, utilize alternative electron acceptors such as nitrat, sulfat, or metal ions, and are often employed for the transformation of halogenate solvents or chlorinated aromatics. Aquatic environments are increasingly subject to diverse anthropogenic contaminants arising from agriculture, industrial, and urban activities, as well as accidentally releases in industrial effluents contribute complex mixture, including heavy metals from metal plating, organic solvent from chemical manufacturing, and synthetic dyes from the textile industry, many of which exhibit high persistence and toxicity. Urbanous water introduces additionally stressors such as heavy metals from stormwater, surfactants from detergents, and emerging contaminants, including pharmaceuticals and personal care products. Accidentally spills, including oil, organic solvents, and cyanide solutions, can produce acute toxicity and localized ecosystem disruption. Algae gels provide a suite of natural remediation mechanisms that mitigate the impact of this pollutant. Key processes include biosorption, is passive binding of metals or other pollutants to cell wall components such as silica in diatoms or extracellular polysaccharides which immobilize toxic ions and prevents future dissemination in the environment. Bioaccumulation is a phenomenon like active uptake of pollutants into the cytoplasm and vacuoles where they can be transformed or stored, reducing bioavailability and trophic transfer. And biotransformation represents enzymatic modification of organic contaminants throughout oxidation, reduction, hydrolysis, or dehalogenation, converting hazard compounds in less toxic or more easily degradable forms. Advantage of using algae from water decontamination. Algae have high bioabsorption capacity, have cell walls with molecules that can naturally bind heavy metals and other inorganic pollutants, quickly capturing contaminants without the need for added chemicals. Bioaccumulation and biotransformation potential. Algae can absorb pollutant into their cells and break down organic contaminants using enzymes, reduce toxicity and removing harmful substances like dyes, pesticides, and pharmaceuticals. Eco-friendly, low environmental impact. Algae treatments avoid creating secondary pollution, protect aquatic ecosystem, and can be integrated with methods of with bioreactors for additional ecological benefit. Microbial degradation of petroleum hydrocarbon is typically evaluated throughout controlled laboratory assay that incubate the hydrocarbon substrate with microbial inoculum under aerobic or anaerobic conditions. The primary objective is to quantify the rate of biodegradation and to understand the metabolic pathways involved. Commonly employed tests and monitoring parameters include respirometric assay, gravimetric analysis, biochemical oxygen demand and chemical oxygen demand, change in indicators, organic indicators, load reduction due to microbial breakdown of hydrocarbons, microbial growth monitoring, microbial population dynamics with optical density or molecular methods to correlate growth and with degradation performance. And enzymatic activity assay when are measured to understand the mechanism base of hydrocarbon transformation. The efficiency of microbial degradation are strongly influenced by environmental parameters that support optimal cell growth and metabolic activity. Controlling this condition is critical for biodegradation potential and pollutant impact. We measure availability of oxygen. Aerobic conditions generally accelerate the degradation of organic pollutants and hydrocarbons, as oxygen serves the terminal electron acceptor in respirometry and enzymatic pathways. Oxygen limitation can slow or inhibit biodegradation processes. Temperature and pH, microbial growth and enzymatic activity are highly sensitive to temperature and pH and pH. Each microbial species has an optimal range. Deviations can reduce metabolic rate or denaturate degradative enzymes. Maintaining suitable temperature and pH ensures maximum biodegradation efficiency and reproducibility in experimental assay. Algal cells, particularly Spirulina platensis, demonstrate substantial potential as biomonitoring agents in ecotoxicological assessments. The studies conducted confirmed that Spirulina platensis not only responds rapidly to toxic stimulus, but also facilitates effective biodegradation of naphthenic acids under controlled laboratory conditions. It's easy of cultivation, rapid cellular growth and metabolic versatile enable the conversion of these organic contaminants in carbon dioxide and water, integrating the biodegradated products into a normal respiratory process. Spirulina can serve as an effective biomonitoring agent due to its rapid response to toxic stimuli. Algae is easy to cultivate and exhibit fast cellular growth under laboratory conditions. The algae show substantial biodegradation of naphthenic acids, converting them to carbon dioxide and water. Combine bioindicator sensitivity with active pollutant mitigation, making it a valuable tool for sustainable water monitoring and remediation. Thank you for attention.
Thank you very much. If someone have question or questions. No. Okay, so we will go to the next presentation. The next presentation is optimization of water supply system through quality monitoring and loss reduction. It's the same university, Petroleum and Gas Ploiești. Who will present? Tot dumneavoastră?
Yes, but one moment, please, because trebuie să caut prezentarea. Numai un pic, vă rog.
Da, nicio problemă. Dacă nu găsiți, o putem da noi.
Am reușit.
Okay, bine.
Trebuia deschisă pe ecran și am...
Da.
Optimization of water supply system through quality monitoring and loss reduction. In the context of increasing anthropogenic pressures, climate change, and the qualitative degradation of water source, the sustainable management and protection on this critical resource have become strategic priorities of both the global and European levels. Drinking water quality monitoring is the systematic process of collecting, analyzing and interpreting physicochemical and microbiological data to verify compliance with legal standards and ensure ongoing consumer safety. This is not just about occasional laboratory tests, but involves a coordinated set of activities, including continuous monitoring of important parameters directly in the water, turbidity, pH, residual chlorine, in the treatment plant to continuous monitoring in the distribution network and at the point of consumption. The data obtained from the basis of operational decisions, allowing the adjustment of treatment process, the rapid identification of deviations, and the implementation of corrective measures. Thus, a robust monitoring program is essential not only for demonstrating compliance with legal requirements, but also for the efficient management of the system. From a water safety perspective, decreases in network pressure due to loss can favor contaminations can become entry points for pollutants into the system. Therefore, reducing losses represents not only an economic efficiency measure, but also an essential component of risk management for drinking water quality. From an operation point of view, losses and pressure variation directly influence the maintenance of the free residual chlorine in the network, which requires careful monitoring and appropriate adjustment of disinfection process. So combining all these aspects reflects the basic idea of drinking water supply system that is both sustainable and able to cope with the challenges for drinking water supply service in line with the requirements of the current European regulatory framework. The surface water treatment process is a classic application of conventional treatment technology as follows the fundamental stage of drinking water treatment. Coagulation, flocculation, this is for This is fourth essential stage on the physicochemical treatment. Depending on the quality on the raw water, particularly the turbidity, the laboratory established the optimal dose of coagulants and flocculants. The system used granular aluminum sulfate. This purpose of this stage is to destabilize colloidal particles and form large flocks that are easily separable in subsequent stages. Decantation is water mixed with the reagents is directed to the radial decanters where the formed flocks are separated by gravitational sedimentation due to the reduced flow rate. Filtration and disinfection is the final and most important stage for ensure the microbiological safety of drinking water. The chlorine dose is established based on the experimental determination of the chlorine breakdown curve. The technological process follows a classic surface water treatment scheme structured in a successive stage of mechanical pretreatment, physicochemical treatment, solid-liquid separation, filtration, and disinfection. Each stage is designed to progressively remove physical, chemical, and biological impurities from raw water, ensuring the production of drinking water that meets the quality requirements in force. The chlorinate system ensures the inactivation of pathogenic microorganisms and maintain a chlorine residual necessary for protecting water in the distribution network. From the tanks, drinking water is pumped in the pumping station and introduced into the supply and distribution system for consumers. Quality or emergency situation thereby ensure the security of the drinking water supply. For water quality monitoring, it is necessary to take sample for previously established point which is critical step in validating laboratory analysis. This etap ensures the representative integrity and traceability of the samples. On this slide, the successive stage of the installation technological process, and examples of accidents and repairs carried out. Free residual chlorine is the key parameter for controlling the disinfection process. Indicate the amount of chlorine available to inactivate pathogenic microorganisms and maintain a protective residual in the distribution network. Residual chlorine monitoring water treatment plan converts the entire technological flow. In the current configuration, the monitoring or filtered water quality is performed globally. Installing individual turbidimeters allow rapid identification on non-performing filter, optimization of washing cycles, reduction of toxicological water and technological water consumption, constant maintain of turbidity below 0.1 NTU. Final disinfection is performed by chlorination. To increase safety and dosage accuracy, it is recommended automatic dose adjustment based on temperature and retention time, simulation of the hydraulic behavior of the supply, estimation of retention time and residual chlorine degradation, detection of deviation from the theoretical model, automatic optimization of coagulant and chlorine dosage. The implementation of numerical hydraulic modeling using quality parameters, accurate leak diagnostics, pressure regime optimization, proactive water quality management. The estimated economic impact on the modernization, the treatment process is suitable, adaptive and correctly sized, and reagent consumption effectively reflects seasonal variation in raw water quality. However, the hydraulic assessment of the water supply relieved the significant structural vulnerability generated by aging pipes with exceeded service lives and by metal section affected by corrosion. Thank you.
Thank you very much. If there are some questions or not.
Yes, actually, I have a question for taking both your presentation. Thank you for your presentation and congrats. I want to ask if you know there are any water treatment facilities in Romania or any interest in implementing them that use algae-based treatment.
I don't know. I don't know. If in Romania is implementing the use of algae in drinking water for people, water for
or for people or for another I don't know, for
I
another
don't know.
purpose.
I don't know. Personal, I make the experiments with algae, but in raw water, in polluted water.
Okay. Thank you very much for your
Thank
presentation
you.
again. Thank you.
Thank
Mă
you.
scuzați, eu am o întrebare dacă e departe. Problema, catastrofa asta ecologică care s-a întâmplat pe Nistru, având în vedere că dumneavoastră sunteți de la Universitatea din Ploiești, Petroleum Gas University, puteți să vă expuneți cu privire la metodele folosite de depoluare a Nistrului, având în vedere că la noi au fost identificate în special hidrocarburi aromatice ușoare, benzen, etilbenzen și se folosesc în special barajele astea adsorbante. Ele sunt eficiente sau nu chiar eficiente? Pentru că eu știu că hidrocarburile aromatice ușoare, ele în mare parte se dizolvă, da, în coloana de apă sau se volatilizează sau cum dumneavoastră credeți? Care este metoda cea mai eficientă? Și într-adevăr, dacă este posibil de folosit ceea ce dumneavoastră a spus, bioremedierea, pentru că într-adevăr capacitatea asta de autoepurare a ecosistemului se bazează pe aceste organisme care sunt destul de eficiente. Iată, pe mine asta mă interesează pentru că este chiar o problemă destul de mare la noi și având în vedere că am ocazia să ascult un specialist.
Da, în măsura în care vă pot răspunde și eu, și noi facem teste, e adevărat, în cazul nostru, în laboratoare, facem teste prin poluare controlată. Adică noi inducem o poluare unei mostre de apă și încercăm să găsim soluțiile cele mai eficiente. Utilizăm și substanțe chimice, utilizăm și substanțe adsorbante și mai nou utilizăm și microorganisme cu rezultate destul de bune. Publicațiile noastre vă pot arăta cam ce am descoperit noi. Acum, la scară largă, sigur că lucrurile nu stau ca în laborator. Pot interveni o serie de factori care, eu știu, pot conlucra împreună. Poate și o altă poluare, poate faptul că apa de care dumneavoastră vorbiți, este foarte important să știm dacă este o apă care colectează impurificatorii aceștia și acumulează undeva sau doar este o apă curgătoare și valul de poluare ar trebui să se diminueze cu trecerea timpului. Deci sunt niște aspecte foarte importante, pentru că una este să depoluăm o zonă unde s-au strâns anumiți poluanți și alta este dacă avem o poluare punctiformă, îi spunem noi, care, eu știu, se diluează cu trecerea timpului. În ceea ce privește poluarea cu hidrocarburile aromatice, da, este destul de severă și este serioasă problema și trebuie găsite soluții, dar numai la fața locului putem decide cam ce ar trebui făcut și, bineînțeles, prin teste succesive să găsim soluția cea mai bună.
Da, vă mulțumesc mult și în primul rând mulțumim părții române pentru ajutorul care l-ați acordat în numele Republicii Moldova. Chiar e foarte, foarte important, având în vedere că pentru noi a fost un test, chiar un test de provocări destul de mari și ajutorul vostru este substanțial. Vă mulțumim mult.
Cu plăcere și la mai mult ajutor dacă putem, cu mare drag.
Mulțumim.
Ok,
Încă o
we'll
remarcă,
go.
dacă s-ar putea, pentru că toți au vorbit în limba română, față de aspectele de poluare, aceleași probleme sunt și în Dunăre, ca și în Nistru, poluări punctiforme. Poate ar fi interesant de făcut in situ ceva proiecte comune, domnul Bulat și doamna Gheorghe.
Sigur.
Cu plăcere, cu plăcere.
Da,
Mulțumim.
poate ne gândim în situații de astea să găsim ca să prevenim, nu poluarea, dar să prevenim, să gândim pașii următori pe care să-i avem repede atunci când ea se întâmplă.
Da, să fim pregătiți.
Ai intrat din nou? Okay, so you can speak, Biplov. It's your time now.
Yeah, so good afternoon, and thank you for giving me that space to share my work with you. So good afternoon, everyone, and honorable delegates. So this is Biplov from Bangladesh. So I'd like to share my work with you. So its title is Empowering Students as Water Stewards, Education for Sustainable Use, Ecosystem Protection and Pollution Prevention. So this is overall outline of my presentation. So basically, all of we know that water is a critical natural resource supporting ecosystem and human livelihood. And globally, we know that water is very important resource, and it is facing many kind of threats like pollution, climate change issues, and unsustainable consumption. Developing countries like Bangladesh and South Asia is actually vulnerable to water-related environmental challenges. And we believe that youth engagement is increasing, recognized as a key driver for environmental sustainability. So the context of the study is Bangladesh faces significant water-related challenges, including pollution, degradation of underground water level, and resource mismanagement. And limited environmental literacy often contribute to unsustainable practices. And educational institution, we believe that can play an important role in developing environmental stewards among young leaders. And many students lack a scientific understanding of water ecosystem and sustainable consumption practices. Traditional classroom teaching often fails to connect environmental knowledge with real-life action. And that's why we believe this model, which is actually experiential and interdisciplinary learning model to empower youth in water stewards. And we, during this study, examined the impact of structured literacy intervention among youth engagement towards sustainability. This program fosters a deeper understanding of water, aquatic ecosystem, pollution control, and stewardship, while strengthening environmental attitude and pro-sustainability behavior. It evaluates changes in student knowledge, attitude, skill, and civic participation following program implementation. So our study is basically focused on 4 objectives, actually to assess the baseline knowledge of the student on the specific context. Second thing is to set up a model which is focusing on experiential and interdisciplinary. And third one, to analyze and evaluate the changes due to this intervention. And finally, the fourth objective was to examine the scalability and replicability of the model. And we focused on 3 testable hypotheses, which is participants who undergo sustainable educational environmental shift model will demonstrate a statistically significant increase in water literacy. And number 2, participant in the working in this model will result in significant positive shift in attitude. And third one, increased literacy on water and aquatic ecology will positively predict participants' commitment to sustainable lifestyle. So we follow actually mixed method study, which is quasi-experimental mixed method pre-post intervention design. And our work was focusing on 6 months of study and a total of 310 participants. And we followed actually 5 steps. So initially we started with baseline survey, then followed by knowledge development intervention, then field visit, and then different kind of competition focusing the theme of the research work, and finally post-event evaluation. So here we can see that step 1, we actually run some survey among the students. What was their baseline knowledge regarding environment, water issue, sustainability, stewardship? And after that, we allowed them to have some kind of experiential training through field engagement, different how water is connected to life, and how are they thinking of the future outline of the water. So through art competition, through debate competition, through field visit, and through some kind of seminar, we try to empower them. And we also added some kind of competition here. The photos here, you can see the participants presented their different kind of outcomes here. So the pollution happening in water issues and how can we address this, they represent in this presentation. And we allow them to develop some kind of eco project, which they are thinking would be better for the environment. So they actually displace this kind of project work. And this work actually helped them to have some kind of behavioral transformation. So knowledge to awareness, then attitude shift, then environmental intention, then sustainable behavior, and finally the stewardship. So this is the study report where we can see that pre and post, there is a drastic change. So the blue line is actually indicating the after intervention and green line before intervention. We can see a significant change in terms of their knowledge in stewardship, sustainability, marine ecology, and aquatic science. And the findings indicate that green lifestyle commitment by 85% of the participants, knowledge improvement, and PR empowerment intention. And before the intervention, they actually did many kind of harmful practices to the aquatic body, which was because of lack of their information. And field-based learning effectiveness was 83%. So the ANOVA comparison shows that field-based learning demonstrates the highest impact compared to the other intervention. And also one important thing is that not all learning experiences are equal. We can see workshop, field visit, case study, idea challenge, they have different level of outcomes. And regression analysis reveals that knowledge gain and field exposure as key predictors of sustainable behavior. And a strong positive relationship exists between higher knowledge leads to better sustainable behavior. And we did some correlation analysis, relationship behavior, that is knowledge is connected to behavior, field learning is connected to awareness, awareness is connected to peer motivation. So we can see strong correlation indicate interconnected growth in knowledge, attitude, and action. And this is a table which indicate that the lifestyle and perception before the program and after the program. So where we can see that before the program, so the participants did many limited access to the different environmental activities, but after the program, they understand the importance, and you can see that their frequency increases. And before the program, you can see the reduction of natural resource consumption was very low, but now they are interested to go for the reducing different consumption of natural resources. So this is the table, which is actually indicating a positive shift of the attitude of the participant. And overall impact is, we can see that 310 participants were directly empowered, and they claim that in their daily that is uses, they are trying to reduce the water consumption, and water pollution by the participants significantly reduced. More than 1,000 were indirectly empowered because these 310 participants are sharing their outcome with the other participants. So this is how we can grow a sustainable stewardship among the youth. The verbatim statement is indicating that the concept of sustainability and stewardship was totally new. My involvement in this journey has opened my eyes, and now I strongly believe that I have a strong role to make our world livable. And another participant, she mentioned that I had very little knowledge on aquatic ecology. In the past, I visited the coastal region of the country, but I believe that my mindset on sustainable natural resource consumption can bring benefit to the whole world. And this is the statement from one mentor. Actually, I came to know that they made many mistakes previously, which caused much harm to the aquatic body, which they did unwillingly and due to lack of knowledge. But now they are very much clear about many issues related to water, sustainability, and stewardship. They want to develop their school community as a steward of the water, not the consumer of the nature. So this is the overall outlook of the model, where we can see that we did some intervention like workshop, online, offline resource sharing, experiential learning, competition, awareness, saving. But we face some challenges like existing schoolwork, parent and school authorities mindset, lack of funding and academic calendars. So this sometimes creates some barrier to reach the exact outcome. But we can see that it creates an opportunity for the students, and it creates an opportunity for protection of biodiversity, reduced consumption, and sustainability. And we can see that it can create sustainability of population, institution, country, particular theme, community, and also the future generation. And we can see there is a scope of scalability of this intervention in terms of youth empowerment, educational institution, in terms of regional, and in terms of global. And our work is actually covered 8 SDG goal: quality education, clean water, sustainable city, responsible consumption, climate action, life below water, life on land, and partnership. And finally, we can conclude that sustainability is the key factor to the considered in almost in all the areas of activity in the coming days, and empowering next generation towards sustainability to protect our aquatic system or ecology can be more impactful. So despite many limitations and challenges, this model reflects that sustainable environmental stewardship can create a significant impact on the youth community. And more partnership and collaboration can make this model more effective in the coming days. And we can replicate this in any part of the globe for the empowerment of the youth. So thank you for giving me the chance.
Thank you very much for your effort to be with us, because also you are between planes, and the time in your country, it's way other comparative with ours. If someone have a question about your presentation.
Yes, hello. Deb, I have one question for you. My name is Marco Ionuț.
Please.
And in recent years, what has been the impact of climate change for Bangladesh? What is the major impact for change climate?
Actually, in southern part of our country, we have coastal communities. So due to salinity intrusion, so the people have to shift their life cycle from coastal area to the urban areas. This is one kind of challenges, and also scarcity of fresh water, drinking water. And it also affecting the agriculture a lot, because lots of freshwater agricultural product nowadays are not able to grow there. And also it is affecting the fishing community there, because you know that if saline water comes to the freshwater body, so it affects the growth of the fish. And also the largest mangrove forest is in Bangladesh, which is Sundarban. So that is also connected to the coastal area. So we can say that due to climate change, the sea level rise, salinity intrusion, and increasing the frequency of different climate-induced natural calamities actually causing harmful to our community.
I understand. Thank you. So same problem to the European zone.
Yeah.
Thank you.
Thank you.
And I have one, because we started this discussion. I know the Bangladesh is mostly the delta. Yeah, we have also the Danube Delta here. It's quite large, but not like Bangladesh one, but it's quite large. And we have there some issue regarding the decrease of the species, decrease of the fish capture, decrease of the environment. Do you have the same problem, because the also quality of water was changed in the last time? Do you have the same problem?
So we have the same issues. So as I mentioned that due to climate issues, so we know that in the coastal part, so water salinity is increasing significantly. It is affecting harm to the that is the species in the freshwater community there, connected to the coastal line. So that's why we are also facing the same thing. And also you can see that continuous rising the mean temperature is also affecting a lot.
And also the rise of the level of sea affect you.
Yes.
Okay.
Yes.
For us, a little bit less like you, but still affect, because we coastal line have severe damage of each storm, and storms start to be growing and growing like power, and this is not good. And the state need to repair this coastline each year. So because mostly there it's tourism, and we want to keep the tourism flow. And but each year the damage grow. Probably it's the same also in
Same here, yeah.
Yeah, now we understand what it's climate change, not local change.
Yes.
Okay. If are not other question, we will go to the next one. Thank you to having you, and we have to go with the others.
Thank you.
We will keep in touch. Thank you for your presence here and for your effort to be with us at this conference. Thank you again.
Thank you.
Next presentation will be a short presentation about climate change implication for fishery and agriculture in Romania. Actually, will be a poster. The presentation will be kept by Ionuț Marcu from Research Development Institute of Aquatic Ecology, Fishery and Agriculture, Galați. Ionuț, you can start.
Hello, good morning, everyone. For who don't know who I am, my name is Marcu Ionuț. I'm the PhD student of the field of the electrical engineering. I represent the Institute of the ICDEAPA Galați from Romania, and today I try to talk by this problem to have our friend Deb to change climate. The topic of this poster is the impact of climate change on fisheries and aquaculture in Romania. The climate change is not a new issue, but its in effect is becoming increasingly visibility to the intense. Rising temperatures, change in precipitation patterns, and extreme events suck to the drugs and flow to the alternative effective aquatic in ecosystem. In the study, we analyzed the climate data, reviewed scientific literature, and evaluated aquaculture system, included both traditional and recirculant system. We also considered observation for Romanian aquatic ecosystem. Our results show that increasing water temperature reduces the oxygen available and the affect fish metabolisms, lending the stress and reducing the productivity. At the same time, drought limit water resource while extreme event can damage infrastructure or reduce fish survival. We also observe change in species distribution and increased risk to the diseases which further impact aquaculture system. The address change challenge several adaptation strategies and the necessary. These include the use of the recirculated aquaculture system, improved management practice, and diversification of the species. In conclusion, climatic change has significant impacts on fisheries and aquaculture in Romania, and the study does present a new problem, but RACH acts the warning signal to this present problem. It highlights the urgency of the taking action to improve the resilience and ensure the sustainability on this sector. Thank you, everyone, and who has some question?
Will be a poster about effect of dietary ulva-lactuca extract of immunological and oxidative stress parameter in common carp exposed to some treatments. Will be to present this paper Alina Nicoleta Macoveiu Dobre.
Hello, everyone. My name is Alina Nicoleta. I'm a research assistant and Research Development Institute for Aquatic Ecology, Fisheries and Agriculture, Galați. We are presenting only a part of my result. So, the use of natural additive in agriculture represent a promising approach to enhancing fish health and therapeutic treatment. Due to the content of bioactive compound, with antimicrobial and antioxidant property. The green macroalgae Vulva lactuca can function as a natural adjuvant and feed of common carp. The effect of Vulva lactuca as a natural adjuvant was evaluated in common carp by administering two experimental diet with and without vulva extract for 11 weeks, followed by treatment with antibiotics, fluorphenicol and tetracycline for 10 days. This approach can serve as a starting point for development solution to limit the excessive use of antibiotic and prevent the emergence of resisting bacterial strains. The design experimental with use a total of 270 common carp with an average weight of about 20 grams. Your use and the experiment. The fish were fed for 11 weeks with two types of diet. A standard diet, a diet supplemented with Vulva lactuca extract. After this period, the fish were randomly divided into 6 experimental groups with 3 replicates per group and 40 fish in each replicate. The group were the first control group, the fish feed conventional diet, the second conventional diet with fluorphenicol treatment, the free group with conventional diet with oxytetracycline treatment, the 4 group diet supplemented with Vulva lactuca extract, the 5 group diet with Vulva lactuca and fluorphenicol, in the 6 group diet with Vulva lactuca and oxytetracycline. The result, we measured the free marker of oxidative stress like activity lysozyme, activity malondialdehyde, and total antioxidant capacity. As you can see, the result indicate the group of vulva had the highest lysozyme activity. The antibiotic group had the lowest. Will Vulva lactuca combined with antibiotic increase activity compared to antibiotics alone? The highest MDA level were found in the control group treated with antibiotics, while the lowest level was observed in the group receiving Vulva lactuca with oxytetracycline, and all group with Vulva lactuca showed lower value than the control or antibiotic only groups. Vulva lactuca extract produce the highest total antioxidant capacity. Antibiotic treatment shows the lowest value, and the combination of vulva with antibiotic had intermediate level, suggesting that vulva reducing the negative effect of antibiotic and has a strong antioxidant effect. And the conclusion, the result obtaining the present study demonstrate the dietary incorporation of Vulva lactuca contribute to the regulation of physiological response by enhancing immune-related parameters and mitigate oxidative damage. This finding highlight the potential of Vulva lactuca as a functional feed additive, the support immune competence and oxidative balance, suggesting its applicability as a sustainable strategy to reduce reliance on antibiotic and agriculture systems. This is of part of my result because the full result to be published shortly. Thank you.
Thank you very much. This was the last presentation of our conference, Water Life Support.